Hello and welcome to Physics Wallah English YouTube channel. I'm Shalini Somashekar, your botany teacher. In today's lecture, we are going to be summarizing the entire chapter of molecular basis of inheritance in one shot. I'm sure all of you are aware of how important this chapter is for your board exams as well as entrance exams like NEET. In the previous lecture, I had discussed principles of inheritance and variation. Both of these chapters are in the same unit. So in the last lecture where we discussed principles of inheritance and variation, we discussed about Mendel's experiments, what were the rules or the laws of Mendel. Do all organisms follow Mendel's rules for all characters? Are there any deviations from it? What are the principles that govern inheritance? What are the various inheritance patterns? What are genetic disorders? All of that was covered in the previous lecture. So in the last chapter, we learned about how DNA controls everything that an organism is. It is responsible for the color of my eyes, the color of my hair, my complexion, pretty much everything that my cells do, DNA is taking control of it, right? So, we already know that DNA is present, the genes are present in DNA and DNA is a part of chromosomes. That much we are aware of from the previous chapter. In this chapter, we are going to learn about how exactly DNA manages to do all of this, right? DNA barely ever gets out of the nucleus in eukaryotic cells unless it's dividing. Despite sitting inside that cozy cabin, how is it able to control all of the things that happen in cells? That's what we're going to discover in today's lecture through the course of this chapter, and the hero of this session is our DNA. So without any further delay, let's get today's session started by talking about DNA. We've already learned about the structure of DNA pretty much in detail in the 11th grade chapter, Biomolecules. So I'm going to quickly summarize all of the different things that you're required to know about DNA in today's lecture. Okay. So DNA, I'm sure you know, is a polymer, right? What is a polymer? Polymer you can think of as a string of beads, right? So many beads joining together to form a string or a chain. You can think of a polymer like that. So polymer is like a long chain, and each bead in this chain is known as a monomer. If DNA is a polymer, then what are these monomers made of? These monomers are made up of units that we call deoxyribonucleotides. Okay? So one nucleotide is joined to another nucleotide by means of chemical bonds that we call phosphodiester bonds. This has been discussed in detail in the biomolecules chapter. If you seem to have forgotten, go back and revise, all right? So, how do you measure the length of DNA? I'm sure you all know that DNA is extremely long molecule, right? The DNA that is present in every one of our cells can be as long as 2.2 meters. That's how long it is. So usually when you're measuring DNA, we measure it in terms of the number of nucleotides or base pairs. DNA is made up of nucleotides, I already told you. Nucleotides are these beads that are joining together to form the DNA molecule. So if you have to measure how long the DNA is, the unit that we usually use is the number of nucleotides that are present in it. If it is a single-stranded DNA, if the DNA is double-stranded, the nitrogenous bases from one strand would have formed hydrogen bonds with the nitrogenous base from the opposite strand. So it would have formed base pairs. If it is a double-stranded DNA, we measure the length of DNA in terms of base pairs. Okay, here are a couple of examples. What we must understand about DNA is that in all organisms belonging to one species, its length is going to be pretty much the same. Okay, for example, if you consider a bacteriophage called 5x174, its DNA has 5,386 nucleotides. This is a single-stranded DNA, which is why we are measuring it in terms of nucleotides. So all of the bacteriophage 5x174 viruses will have the same number of nucleotides unless there is any mutation or unless there is some defect in the virus. Similarly, there is another virus called bacteriophage lambda, which is also double-stranded. In this virus, you can find 48,502 base pairs. I'm sure every one of you have heard of E. coli. E. coli is a bacterium that resides in our gut, in our intestines. This bacterium has DNA that is double-stranded, and the number of base pairs here is 4.6 into 10 to the power of 6. These are important numbers that you have to remember, memorize for your entrance exams. Okay. So, if you consider the haploid content of human DNA, just one set of chromosomes in human DNA, we have 3.3 into 10 to the power of 9 base pairs. That's how long one set of human DNA is. All right. So these are certain examples you have to keep in mind. What I mean, why this is important is because you can see how across different organisms belonging to the same species, this number is pretty much maintained as it is. There's not a great deviation from this number. All right. So that is the characteristic of an organism. Now let's move on and talk about the structure of a polynucleotide chain. I already mentioned in the previous slide that DNA is a polynucleotide. It is a polymer of nucleotides, so we call it a polynucleotide. What are the individual monomer units called? They are called nucleotides. So the monomer is called nucleotides. This is a mistake. It's not a monosaccharide. It should be a monomer. Okay. So the monomer is called a nucleotide and the polymer is called a polynucleotide. You can see how each monomer is joined to the next monomer nucleotide by means of a chemical bond that we call the phosphodiester bond. How exactly this is formed, we'll get to it later. But for now, just remember, it's formed. They're linked together by means of phosphodiester bonds. Now, let's look at an individual monomer unit, which we call a nucleotide. So if you can think of it as a bead, each bead has 3 distinct components. The first component is a sugar, which has 5 carbons. So we call it a pentose sugar. I'm sure you know that penta relates to 5. So there is a pentose sugar. The second component is a nitrogenous base. And the third component is a phosphate group. So you have a sugar, you have a nitrogenous base, and you have a phosphate. Here's how they are attached to each other in a nucleotide. At the center, you will have the pentose sugar. Here, you have the pentose sugar. The pentose sugar will have 5 carbons. Let's start counting these carbons in clockwise direction. Here will be our first carbon, second carbon, third carbon, fourth carbon, fifth carbon. Fifth carbon will be present outside the ring. To the first carbon of the pentose sugar, the nitrogenous base will be attached. This bond is called a glycosidic bond. To the fifth bond of the pentose sugar, the phosphate group will be attached, and this bond is called the phosphoester bond. So basically, in a nucleotide, you have 3 components. At the center, you have a sugar. To its left-hand side, it is holding on to a nitrogenous base. On its right-hand side, it's holding on to a phosphate group according to this illustration. But it can be the other way around too. We'll look at how that is a little later. Okay? So this is what are the different components that you can find in a nucleotide. Now, you know that in some organisms, some viruses, RNA also can be the genetic material. And RNA can either be single-stranded or double-stranded and act like a genetic material. We know that RNA also is a nucleic acid. RNA also is a polymer. RNA and DNA have many similarities. They also have several differences. Now let's talk about one such difference between RNA and DNA. RNA and DNA both are polymers of nucleotides. So they're both made up of a string of beads. But each nucleotide also has 3 components in both of them. But the sugar that is present in the center is different in DNA and RNA. In both RNA and DNA, we have a 5-carbon monosaccharide. All right. In RNA, we call it ribose. We call it ribose. And in DNA, we call it 2'-deoxyribose or simply deoxyribose. Now look at this illustration and try to find out what exactly is the difference present between these two molecules. They're both sugars, they're both having 5 carbons. The difference lies in the second prime carbon. Look here, I said 2'-deoxyribose. The difference between ribose and deoxyribose lies in the second carbon. Let's first begin numbering these carbons from this first carbon in clockwise direction. So here is our first carbon, here is our second carbon, here is our third carbon, here we have our fourth carbon. Like I already mentioned, the fifth carbon is outside the ring and you can find it here. Let's do the same thing over here as well. Here we have carbon 1', carbon 2', carbon 3', carbon 4', and carbon 5'. Now let's focus only on the second prime carbon, which is this carbon here. As you can see, in ribose, attached to carbon, you have a hydroxyl group. Over here, we have a hydroxyl group. Whereas when you look at deoxyribose to the same second carbon, you have a hydrogen attached instead. Here you have OH, here you have only H. Here O is removed, which is why we call it deoxy. Deoxy, oxygen is removed. Here you have OH, here you have H. So this is called deoxyribose. Except for that one difference, everything else remains the same, both in ribose as well as deoxyribose. There's no other difference, absolutely. One more thing I want you to understand and remember at this point is, why do we number it as prime? 1', 2', 3', and not simply 1, 2, 3. Remember I said every nucleotide has 3 components. You have the sugar. Let me use a different color. Yeah. So we have a sugar, we have a nitrogenous base, and we have a phosphate group. This nitrogenous base also has carbons. This sugar also has carbons. So when I say the first carbon of a certain nucleotide, would you think of the carbon that is present in the sugar, or would you think of the carbon that is present in the nitrogenous base? That would become extremely confusing. So in order to avoid that confusion, while numbering the carbons that are there in the sugar, we add a 1', 2', 3'. This dash that you add on the right top as a superscript for these numbers, that is read as 1', 2', 3'. Okay. So that will differentiate and reduce the confusion that is there when you're numbering the carbons in the nucleotide. So in the carbons in the nitrogenous base are numbered 1, 2, 3. Here it is. In the sugar, it is 1', 2', 3'. That is basically done to avoid any confusion. Now, sugars, like I already mentioned, is the central molecule in a nucleotide. Now, let's talk about the second component in a nucleotide, which is the nitrogenous base. What are nitrogenous bases? They are heterocyclic rings. Within these rings, definitely you will find nitrogen, which is why it's called nitrogenous bases. Apart from nitrogen, we will also find carbon, oxygen, and hydrogen. Okay? So, these nitrogenous bases are basically of 2 kinds. The first type is called purine and the second type is called pyrimidine. Can you look at these structures and find out what is the difference between purines and pyrimidines? Forget about their names and just observe their structures. It's very evident that the purines are larger in size compared to pyrimidines because these purines have 2 rings in their structure, whereas pyrimidines have single ring. The name purine is short, the word is short, but the size is big. The name pyrimidine is big, but their size or structurally they are small compared to the purines. So this is the difference between purines and pyrimidines. You find both kinds of nitrogenous bases in both DNA and RNA. Now let's talk about purines. Purines are of 2 kinds. You have adenine, which we abbreviate as A. We have guanine, which is abbreviated as G. Both of these are double ring structures, purines. So, pyrimidines are of 3 types: cytosine, thymine, uracil, that are abbreviated as C, T and U respectively. Over here, We are talking about both DNA and RNA, and I already told you what is the difference with respect to the pentose sugar. Over here, even in the nitrogenous bases, you can find difference between DNA and RNA with respect to the nitrogenous bases. Adenine, guanine, cytosine. All of these are found both in DNA as well as RNA. These three are common to both DNA as well as RNA. Thymine is found only in DNA. Uracil can be found only in RNA. So basically, the nitrogenous bases that you find in DNA include adenine, thymine, guanine, cytosine. The nucleotides that you find, or the nitrogenous bases rather, that you find in RNA are adenine, uracil, guanine, cytosine. I hope this is clear. Now let's move on and talk about what nucleosides are. I already mentioned that regardless of whether it is DNA or RNA, you will have a pentose sugar. To the first carbon of this pentose sugar, prime 1 carbon, 1 prime carbon, nitrogenous base is attached by means of a glycosidic bond. This molecule where a sugar is attached to a base is called a nucleoside. Understand very carefully, nucleotide has 3 components, nucleoside has only 2 components. If you remove the phosphate from a nucleotide, what you get is called a nucleoside. So nucleosides will only have sugar and a nitrogenous base. Now, depending upon which is the nitrogenous base present, we call it different names. If adenine is present, we call it adenosine. A nucleoside containing adenine is called adenosine. A nucleoside containing guanine is called guanosine. If it contains cytosine as the nitrogenous base, we call it cytidine. If it contains thymine, we call it thymidine. If it contains uracil, it's called uridine. If it's RNA, we just call it with these names. If the sugar here is deoxyribose, then we call it deoxyadenosine, deoxyguanosine. You just add the prefix deoxy to indicate that the sugar there is deoxyribose. Alright, now let's move on and talk about, just let's take a look at the structures, different structures of the RNA nucleotides in the first row and the DNA nucleotides in the second row. You can see how the only difference here with respect to the sugar is in the second carbon, and instead of uracil, you have uridine in DNA, but otherwise they're pretty much the same, right? Now let's move on and talk about the third component, which is the phosphate group. This is the only thing where there is no difference between DNA and RNA. In both cases, it is PO4 minus. So this phosphate group is attached to the fifth carbon, which is present outside the ring in both DNA as well as RNA. So that's about the phosphate group. Now let's talk about the nucleotide and its nomenclature. In a nucleotide, you will find all the 3 components. Centrally, you have a sugar. You have, on one hand, it's holding on to a nitrogenous base. On the other hand, it is holding on to a phosphate group. If the nucleotide contains adenine and a phosphate group as well, so it's a nucleotide containing all the 3 components, it is called adenylate acid. If it contains G, it's called guanylic acid. If it contains C, it's called cytidylate acid. If it contains T, it is called thymidylate acid. If it contains uracil, it's called uridylic acid. Again, if they are DNA nucleotides, then you add deoxy as a prefix. Alright, so this is about nucleotides. Let's take a look and understand how a nitrogenous base, a nucleoside and a nucleotide differ from each other. Here is an example that I've taken of adenine. What is adenine? Adenine is a nitrogenous base. Adenine here attached to a pentose sugar will give us adenosine, which is a nucleoside. To this nucleoside, at the 5th prime carbon, if you attach a phosphate group, you will get adenylate acid, which is a nucleotide. Nitrogenous base plus sugar, nucleoside. Nucleoside plus phosphate, nucleotide. Nucleotide minus phosphate, nucleoside. Nucleoside minus sugar, nitrogenous base. I hope this is clear. Now let's move on and talk about how exactly phosphodiester bond formation takes place. Where is phosphodiester bond taking place? It is forming in between two nucleotides. Okay, so let's consider two nucleotides over here. We have one RNA nucleotide here. Let's number it. This is the first one. Here we have the second one. The bond formation happens between the hydroxyl group that you have in the 3rd prime carbon of the first nucleotide and the hydroxyl group that you have in the phosphate group attached to the 5th carbon of the second nucleotide. So there is a condensation reaction that takes place and a water molecule gets eliminated. As a result, whatever bond formation happens, this bond here is what a phosphodiester bond is. Is that clear? As a result of this reaction, a molecule of water gets eliminated. Where exactly is the bond happening? It's happening between two hydroxyl groups. It is happening between the hydroxyl group attached to this 3rd carbon in the first nucleotide and the hydroxyl in the phosphate attached to the 5th carbon of the second nucleotide. Between the 3' carbon and 5' carbon, a bond is forming. That is what is a phosphodiester bond formation. The same thing goes on. Below this, there will be the 3rd nucleotide and the OH of the 3' nucleotide will form a bond with the OH in the phosphate of the 5' carbon of the 3rd nucleotide. As a result, the chain will go on extending and increasing in length. Alright, so that's how phosphodiester bond forms. Now here you have a polynucleotide chain that is shown. Here you can see that there are 1, 2, 3, 4 nucleotides represented. This is our first nucleotide. Here we have our second nucleotide. Here we have the 3rd nucleotide. Here you have the 4th nucleotide. Over here, I want you to observe and notice the relative positions of sugar, phosphate and nitrogenous base. Alright. So let's go on writing here. This is our phosphate group. Here we have the sugar. This is our phosphate group. Here we have the sugar. You have a phosphate here and a sugar here. Eventually, there will be a phosphate here and a sugar here. Towards the center, projecting inward, you have the nitrogenous bases, right? So here you have nitrogenous base, here you have nitrogenous base, nitrogenous base, nitrogenous base. So if you look at this polynucleotide chain, it is very evident, very clear that the backbone is made up of phosphate and sugar, phosphate and sugar, phosphate and sugar alternating with each other. From this backbone, the nitrogenous bases are projecting inwards. Alright, so this is how it is in DNA. You all know about this ladder, right? Everybody is aware of this. In every toothpaste ad, in every, hand wash ad or a sanitizer ad, you will see these images popping up. So this is DNA. DNA is like a ladder. It has two strands that are pairing. Over here, the backbone of this ladder is made up of sugar and phosphate that are alternating with each other, and whatever is facing inside, these are the nitrogenous bases. So the backbone is composed of sugar and phosphate, and the nitrogenous bases project inward from the backbone. Alright, now let's talk about how exactly we know about the structure of DNA. How did we come to find the structure of DNA? Today, it's very easy for us to say that DNA has this and it has a ladder-like structure. It looks like a twisted ladder. It looks like a spiral staircase. All of that we know. But how did researchers find that out about DNA? Because if you remember, when we spoke about principles of inheritance and variation, there are multiple times in the textbook where they say, okay, so back then they didn't know that DNA is the genetic material. They did not know, they did not have a physical proof for DNA or factors, as Mendel called it, and they were not aware then. As and when microscopy developed and evolved over time, people were successfully able to elucidate the structure of DNA. What are some of the experiments that people performed? Let's talk a little bit about it, starting with the work of Friedrich Nietzsche. Friedrich Nietzsche is the first person to have identified DNA as an acidic substance. DNA is acidic definitely because it has that phosphate group. Because of the phosphate group, it is acidic in nature. The first person to have identified that feature of DNA is Friedrich Nietzsche, and since it was present in the nucleus, he called it nuclein. This happened when? In the year 1869. Eventually, two people, Maurice Wilkins and Rosalind Franklin. Rosalind Franklin is celebrated as one of the most phenomenal women scientists, researchers in the field of biology. I think you should go and read up a little bit about her. It's a very interesting journey. So what basically they were working on, Maurice Wilkins and Rosalind Franklin, was X-ray diffraction. They were performing X-ray diffraction of DNA, and they found out that DNA has two strands, and they already said that it's helical in nature. Okay, so they already said that DNA has two strands and it is helical. Then we have Irwin Chargaff. Irwin Chargaff, he also studied double-stranded DNA, and he said that in a double-stranded DNA, adenine thymine ratio and guanine cytosine ratio are constant and it is equal to 1. So what that basically means is, he said that adenine, the number of adenine nitrogenous bases is equal to the number of thymine nitrogenous bases. Guanine number is equal to the number of cytosine. The ratio of A and 2, the ratio of G and C is equal to 1 is what he said. And that is exclusively in double-stranded DNA. Based on Irwin Chargaff's and Maurice Wilkins and Rosalind Franklin studies, two people, James Watson and Francis Crick, proposed their famous Watson and Crick double helical model to explain the structure of DNA. They came up with the idea that the two strands of DNA, there is pairing between nitrogenous bases. They said there are two strands. These people said A is equal to T and G is equal to C. Based on their studies, they said that there is pairing that is occurring between the nitrogenous bases in one strand and the other strand. Okay, now let's move on and learn about what exactly are the salient features of the double helical model of DNA that was proposed by Watson and Crick. Clearly, it is made up of two nucleotides, two polynucleotides, right? That is why it's called double helical. The backbone, like we already know, is composed of alternating sugar and phosphate units, and the bases project inside. The two chains have anti-parallel polarity. This is one important thing that you'll have to remember and also understand very well. So what happens here is, so here there are four nucleotides that are given. We have 1, 2, 3, 4. At the end of the first nucleotide, to the 5th prime carbon of this sugar, a phosphate group is attached. On the other end of the same polynucleotide chain, you can see at the free end, this is the free end of this nucleotide. At the free end, you can see at the 3 prime carbon, there is a hydroxyl group. In a single stranded DNA or in a strand of DNA, at one end, one free end, you will have a phosphate group. On the other free end, you will have a hydroxyl group. This phosphate group is attached to the 5th prime carbon, so we call it the 5 prime end. This hydroxyl group here on the other end is attached to the third prime carbon, so we call it the 3' end. So here in one strand, on one side you have the 5' end, on the other side you have the 3' end. Now let's take a look at the other strand. This is the first strand, this is our second strand. In the second strand, you can see how this entire thing is ultafied. It's like inverted. Wherever you have the phosphate group in one strand, on the same side in the other strand, you will have a hydroxyl group attached to the 3' carbon. On the opposite side of the same strand, you will have a phosphate group attached to the 5 carbon, because here this is ultafied or it is inverted. Why should that happen? Because nitrogenous base pairing should happen. If they are both present in the same direction, nitrogenous base pairing will not be able to take place. Therefore, these both are anti-parallel in nature. If one of the strand is running from 5' to 3', the other one will be from 3' to 5'. So if there's 5' of this strand here, opposite to it, on the other strand will be 3'. If there is 3' end on one strand here, on the opposite strand there will be 5'. They are parallel but opposite in polarity. Therefore, we say it is anti-parallel in nature. One chain has the polarity 5' to 3', the other one has it from 3' to 5'. So this is what anti-parallel means. Now, let's talk about the nitrogenous base pairing. The nitrogenous bases from one strand form hydrogen bonds and pair with the nitrogenous bases on the other strand. So, hydrogen bonds are formed. What's important to note here is that any nitrogenous base cannot form pairs with any nitrogenous base that it likes. C cannot be like, I want to pair with A today, tomorrow I want to pair with T, day after tomorrow I want to pair with G. It doesn't work like that. There is extreme specificity for base pairing that we call complementarity. If you've noticed, whenever we draw DNA, there's no variation in its width. Throughout the length of DNA, it's of equal width, it's of uniform width. That is maintained because every time a purine pairs with a pyrimidine. Purine has 2 rings, pyrimidine has 1 ring. So if a purine comes with a pyrimidine each time, the width will be maintained. What will happen if 2 purines pair? They are both bigger in size, that portion of the DNA will become wider. What if 2 pyrimidines decide to pair? They are both small, that portion of the DNA will become narrower. That is not how DNA is. DNA has uniform width throughout its length. That is maintained because of this complementarity, where a purine always pairs with pyrimidine. Another thing is, any purine cannot pair with any pyrimidine. There is specificity in that as well. So, adenine will always pair with thymine by means of 2 hydrogen bonds. Guanine will always pair with cytosine by means of 3 hydrogen bonds. That is why in Chargaff's rule, we saw how the ratio between A and T, G and C is always equal. That is because how many ever adenines are there in a double-stranded DNA, that many thymines would have paired opposite to it. How many ever guanines are there, that many cytosines would have paired opposite. Okay? So here you can see how you have C paired with G by means of 3 hydrogen bonds. In one strand you have T, the other strand you will have A. If you have G here, you will have C here. If you have T here, you will have A here. That is how beautifully they pair. Okay? So this is like Rab ne bana di jodi. So that's about the base pairing. Okay. Now, we say that DNA is helically coiled. We say that it's like a spiral staircase. It's like a twisted ladder. So, if it's twisted, that's obviously helical. There is a specific direction in which bDNA will turn around or coil. Okay. There are so many different kinds of DNA. What we mostly find and what we mostly study about is called bDNA. That's all you'll have to remember. Forget about the other types. These 2 chains are coiled in a right-handed fashion. Now, what do we mean when we say right-handed fashion? Hold your right hand, fold it like this. This is like the axis. Your thumb is like the axis. The DNA will coil in the direction of your other fingers. That is what we mean when we say it coils in a right-handed fashion. The pitch of the helix is 3.4 nanometers. What is a pitch? one complete turn, one full 360-degree turn is called a pitch, or sometimes we even call it as the gyre. So that will measure about 3.4 nanometer, or if you're measuring it in terms of angstrom, it'll be 34 angstrom. It's best if you remember these numbers in nanometers, because that's how it is given in NCERT. Okay. So in one pitch, one full turn, there are roughly 10 base pairs. So in this one full turn, you can find about 10 base pairs. If one pitch is measuring 3.4 nanometers and here you can find 10 base pairs, what will ideally be the distance between two consecutive base pairs? That would be 3.4 divided by 10, which is equal to 0.34 nanometers. So the distance between two consecutive base pairs in a DNA is approximately 0.34 nanometers. Now, in this double-stranded helical DNA, the plane of one base pair will stack on top of another. That will provide it stability and allow it to perform its job more efficiently. Okay, so that's DNA. We saw how the width of DNA, okay, this I forgot to mention, the width is 20 angstrom or 2 nanometer. This width is uniform throughout the length of DNA. We also saw how the DNA length can be very long, and how I said it's the haploid DNA content in human itself will measure 3.3 into 10 to the power 9 nanometers. I'm sorry, 9 base pairs, right? So that's how lengthy it is. So if you take out the DNA from our cell and measure it, it can go up to 2 meters or greater than 2 meters. But our cells are super tiny. The cell itself is measuring in microns. The nucleus within the cell is even tinier. Within that, how is this amount of DNA, this long thread of DNA able to fit? It cannot fit unless it compacts itself. For example, let's take a thread and a roll of thread. You know the thread that they use for sewing? Unwind that thread and you can take it for kilometers, right? But if you take a cardboard roll and roll it around it, start winding it around that cardboard roll, you'll get a compact structure, which is exactly what our cells also do. In order to reduce the length of DNA and make it more easier to access and avoid it becoming messy, they will start winding this DNA, which is very thin, around certain proteins, and those proteins will make the length of DNA thick, but its width will increase. From 20 2 nanometer, it will increase, go on increasing, but its length will start decreasing as it becomes compact. So how exactly does this packaging of DNA take place in prokaryotes? Let's understand that first. In prokaryotes, which include bacteria, always DNA, regardless of prokaryotes or eukaryotes, DNA is negatively charged. This negatively charged DNA, with the help of some proteins that are positively charged, they will become compact and they will lie in a region called nucleoid. Remember, prokaryotes do not have a nuclear membrane. So the DNA, along with certain positively charged proteins, is present in a region that's called nucleoid. So within this nucleoid region, DNA is organized in large loops and that is held by proteins. Now let's talk about how packaging happens in eukaryotes. This is really interesting. So in eukaryotes, we have special type of proteins that are involved in compaction or condensation of DNA. If you remember, when we talk about mitosis and meiosis, there is one phase called prophase where we speak about how DNA gets condensed, right? So this is exactly how it happens. So the proteins that I'm talking about are called histone proteins. Since DNA is negatively charged, the histones have a positive charge. What gives proteins their positive charge? They get it from their amino acids. These histone proteins are rich in amino acids that are basic in nature, such as lysine and arginine. These amino acids carry a positive charge in their side chain. Therefore, the overall protein will be positively charged and basic in nature. There are 5 such histone proteins. You don't have to worry about them. 4 of them in multiples of 2 will form a structure. They will come together, 8 of them, and form a structure called a histone octamer. Octa means 8. So it's called the histone octamer. Around this histone octamer, the DNA will coil around. You can see it here. This is our naked DNA, which is about 2 nanometers. 1, 2, 3, 4. Here you can see 4. There will be 4 behind. This is the histone octamer. Around this histone octamer, you can see how the DNA will bind. So from 2 nanometer, its width will increase to about 10 or 11 nanometer. Different books give you different numbers. In some it says 10, in some it says 11. Okay, so that is winding around, DNA winding around histone octamer. This structure where DNA is wound around a histone octamer is called a nucleosome. So this is a nucleosome, this is a nucleosome, this is a nucleosome. You can see how they all look like a string of beads, right? So what is a nucleosome? The negatively charged DNA that gets wrapped around the positively charged histone octamer, that is the nucleosome. Roughly, it contains about 2000 base pairs of DNA. This repeating unit, like I said, it's repeating. Nucleosome after nucleosome, it's repeating. This is called chromatin, and you can find it inside the DNA, inside the nucleus. Remember, we call it chromatin, chromatin getting condensed into chromosomes. We use all of these terms when you're talking about cell division, right? So this DNA binding around histone proteins and forming nucleosomes attached to each other, that is called chromatin. And like I already mentioned, they're seen as beads on string when you look at it under the electron microscope. So eventually, with the help of more proteins that are called non-histone chromosomal proteins, or simply NHC proteins, they eventually become extremely compact and form the chromosomes. So within chromosomes you have DNA. Within chromosomes you also have proteins that have helped this DNA to become condensed. So chromosomes are not entirely DNA. Okay? So that's about compaction. Now I said over here that the repeating units in of a structure in a nucleus is called the chromatin. This chromatin can be of 2 types. One is called euchromatin and the other one is called heterochromatin. In our DNA, every segment of the DNA will not carry information. There are some segments in the DNA where there is no information. There are some segments in the DNA where there is a lot of information. So wherever there is information, because the information has to be transferred, in such regions, the DNA will be relatively a little loosely coiled, and wherever there's not much information, over there, you can see that it is tightly coiled. Whichever regions have information, that is called the euchromatin, that will be loosely coiled. Whichever doesn't have that much information will be called heterochromatin, and they are relatively tightly coiled. When you add a stain or a dye while visualizing these things under the microscope, euchromatin takes up less stain, heterochromatin takes up more stain and appears darker. These are transcriptionally active. Transcription, what exactly that means, I'll tell you eventually. That is a way, one of the steps in how information gets transferred. So they are transcriptionally active, and because this doesn't have any information in it, these are transcriptionally inactive. Usually the information in heterochromatin is not required a lot of times, or as frequently, that is why it will be very tightly coiled. Here there will be information present that will have to be expressed and transferred all the time. Therefore, there it will be a little loosely coiled. That's about heterochromatin and euchromatin. Okay, so now we know about the structure of DNA. We know about how people found out that DNA had two strands, it had base pairs, A always binds with T, G always binds with C. How they all found out about all of this, we know now. But how did people find out that DNA is the genetic material? There were many people that performed many experiments to find out what exactly is the nature of the genetic material. All of our cells have proteins, carbohydrates, lipids, RNA, DNA, anything could have been the genetic material. How did people conclude that it is DNA itself that is transmitting characters from one generation to another? So there are a couple of interesting experiments that we'll have to learn. All right. So let's begin with Griffith's experiment. It's also called the transforming principle experiment. This is a very interesting experiment, very easy also. So this was performed by a person called Frederick Griffith. That's why it's called the Griffith's experiment. So what basically he does here is he uses two organisms. One is a mouse or mice. Another one is a bacterium called Streptococcus pneumoniae. Streptococcus pneumoniae. This is the bacterium that caused pneumonia in mice. So he was studying these bacteria. And he knew that this bacteria existed in two strains. One he called the rough strain or the R strain. The other one was called the smooth strain or the S strain. What are strains? Just like you have breeds in dogs, you have pug, you have a Labrador, you have a German Shepherd, you have a Golden Retriever. All of these are dogs, but they're of different breeds. They're of the same species. Similarly, in microbes like bacteria, some species have more than one strains. The difference between rough and smooth is that the colonies of rough bacteria, they appeared rough because of the absence of capsule around it. In smooth bacteria, the capsule was present. So smooth strain bacteria were encapsulated, rough strain bacteria were not encapsulated. Rough strain bacteria did not have the ability to cause the disease, so we call it non-virulent. The smooth strain bacteria had the ability to cause pneumonia, so they were called virulent strain. Okay, so this is something you have to remember. Remember, mice, streptococcus, two organisms used in this experiment. So what he basically did was he took a couple of mice. First, he took the R strain bacteria, which did not have the ability to cause disease, and injected these bacteria into the mouse's body. Clearly, because it does not have the ability to disease, it did not affect the mouse at all. Mouse happily lived, survived. Okay. Next, he took the smooth strain bacteria, which had the ability to cause pneumonia. He injected them, sadist, into the mice. Because it had the ability to cause the disease, the mouse got pneumonia and it died. Next, what he did was, he took the S strain bacteria, he killed them by applying heat. So he heat killed these S strain bacteria. Bacteria are dead now because of the application of heat. So he took these heat killed S strain bacteria and introduced them into the mice. What do you think would have happened? The mouse would have survived, the mice would have survived, because the bacteria, when it's alive, it has the ability to kill. But once you kill them, they cannot cause disease. So these bacteria could not cause disease in the mice, and the mice survived. Now let's look at the first case and the third case. The first and the third. Rough strain, mouse survived. Heat killed smooth strain, mouse survived. What he did next was, he mixed this and this together in the next set of experiments. So he mixed them together rough strain and heat-killed smooth strain and injected that into the mice. What do you think should have happened, or what would we have expected? Since individually both of them were not able to cause disease, the bacteria could not have caused disease, the mouse should survive. But to his surprise, the mouse developed pneumonia and it died. So after the mice died, he performed an autopsy and he found in the mouse's body live S strain. He introduced heat-killed S, he introduced live R, but he got live S. How could it have happened? What are the two possibilities? Either the heat-killed S has come back to life and become live S, or possibility 2, live R for some reason has transformed into live S. The first possibility cannot happen in real life. Somebody that has died cannot come back to life. That can happen only in TV serials, not otherwise. So here you can rule out that possibility. What else could have happened? The R strain could have transformed itself and become the S strain. So what's the basic difference between an R strain and an S strain? The presence of the capsule. S strain has the capsule, R strain did not have the capsule. So from this observation, what Frederick Griffith concluded is there is something that is present in the heat-killed smooth strain bacteria that is transforming the R strain bacteria, live R strain, to live S strain. Something from the heat-killed smooth strain is responsible for this transformation. He does not say if it's a protein, he does not say if it's RNA or DNA or carbohydrates. He does not give the chemical nature of what is the thing that is transforming it. But he says there's something in the heat-killed S that is capable of transforming live R into live S, and he calls it the transforming principle. That's why it's called the transforming principle experiment. Alright. So this is basically what we found out from Griffith's experiment. Some people took Griffith's experiment to the next level and performed some more experiments with the same principle basically. Okay, so this is called the biochemical characterization of the transforming principle. They went on to find out what could it be. Could it be DNA? Could it be RNA? Could it be proteins? What could be the nature? So 3 people worked on this. Their names are Oswald Avery, Colin MacLeod, Macklin McCarty. So these 3 guys worked on it. What they basically did was, we know for sure that something in the heat-killed bacteria, heat-killed S strain bacteria, are what is responsible for the transformation. So he takes the heat-killed S strain bacteria. From this heat-killed S strain bacteria, they isolate sugars, RNA, proteins and DNA. Lipid is not even in the race. Nobody even thought that lipids could be the genetic material. So the main contenders for who could be the genetic material was what could be the genetic material were carbohydrates, proteins, DNA and RNA. Most people believed that proteins were the genetic material. Okay. So what they did was they took the heat-killed S strain bacteria, they separated out sugars, proteins, RNA and DNA from them. To these extracts of sugar, RNA, protein and DNA, they added live R strain bacteria. So when live R strain bacteria were added to all of these test tubes extracts, they did not find transformation in test tubes where sugar, RNA and proteins were there. Wherever DNA was there, in whichever test tube DNA was there, over there they saw transformation of live R strain into live S strain. Excuse me. So transformation means, you know, right? You've seen these transformation videos on Instagram and YouTube and all of that. Somebody comes there and they do this and go away and they're a different person altogether. So that's what happened here basically. So R strain drank this DNA and then it transformed into S strain. So S strain, that's how this transformation happened. And this kind of confirmed that it is DNA that is responsible for the transformation of R strain into S strain bacteria. Okay. So still people had their doubts. So they performed another similar kind of experiment with the transforming principle. They took the heat-killed estrine bacteria. From that, they removed the lipids and sugars, because I told you the primary contenders were proteins and nucleic acids. Whether it was protein, whether it was RNA, whether it was DNA, that is what they wanted to find out. So they took this extract and they removed the sugar and lipids from it. To these extracts, they added protein digesting enzyme called protease. They added RNA digesting enzyme called RNase, and they add DNA digesting enzyme called DNase. To these, they added the live R strain bacteria. In whichever test tubes protein digesting, RNA digesting enzymes were there, estrine bacteria appeared. In whichever test tube DNA digesting enzymes were there, S cells did not appear, because DNA was broken down. If DNA was broken down, transformation is not possible. That indicates that DNA indeed is the genetic material. So after this, two other people, Alfred Hershey and Martha Chase, performed experiments to give the final proof that DNA itself is the genetic material. What exactly was their experiment like? They made use of two organisms. One is a virus, the other is a bacterium. Viruses that infect bacteria are called bacteriophages. So they used bacteriophage, a bacteriophage that infects the E. coli bacteria. So which are the two organisms that they made use of? Bacteriophage and E. coli. This causes infection. This virus causes infection in E. coli bacteria. So what they basically did here was, they did radio labeling. They performed radio labeling of certain components in viruses. They took these bacteriophages. In bacteriophages, in viruses, you have two components. You have an outer protein coat. Inside that, you have the genetic material DNA. Now we have to find out, is the protein the genetic material or DNA the genetic material? Protein will contain amino acids like methionine that contains sulfur. In DNA, there is no sulfur. In protein, you do not find phosphorus. In proteins, you do not find phosphorus. In DNA, phosphorus is present in the protein coat. In the phosphate group. So phosphorus is present. They took two sets of bacteriophages. In one, the protein coat was radio labeled. See here, whatever I am drawing in red, that is radio labeled. So they took a radioactive isotope S35 to grow these bacteriophages. So the protein coat was radio labeled. In the other set of bacteriophages, DNA was radio labeled with P32, the radioactive isotope of phosphorus. They allowed these two sets of bacteria to infect E. coli bacteria. In the first step, which is called infection, they allowed this to infect it. Right now, we know that the DNA is going inside, protein is staying outside. Back then, they did not know. Okay. So what they did was, they took bacteriophage and they inserted that, or they used that to infect the E. coli bacteria. What they did next was, with the help of a blender, they removed the protein coat. If you remember the life cycle of a virus, the genetic material gets injected inside the host cell and the protein coat remains outside. So they wanted to separate the protein coat from the bacterial cells with the viral genetic material. So they spun it in a blender. So all of these protein coats got separated at that point. Next, they performed centrifugation. Centrifugation is similar to blending, where you're rotating these inside of centrifugation tubes at very high speeds, like 50,000 rotations per minute, 25,000 rotations per minute, very high speeds. So when you're rotating it at such high speeds, The bacteria being heavier or denser compared to a viral protein coat, it will form a pellet at the bottom of the centrifuge tube, and the supernatant will remain on top, and the supernatant will contain the viral protein coat. That is exactly what happened. Now, if you see, the bacteria itself was not radiolabeled. Two sets of bacteriophages were radiolabeled, right? If you see, after they tested for radioactivity, in whichever set of experiment, DNA of the bacteriophage was radiolabeled. In that, in that set, in the bacterium also you could find radioactivity. In the other set where the protein coat was labeled, over here, the bacterium did not show any radioactivity. The supernatant showed the radioactivity. Over here, the residue showed the radioactivity. Since the bacteria itself wasn't labeled, if the bacterium is showing radioactivity, that indicates that something from the virus should have gotten inside the bacterial cell, and that is what is causing the bacterium to become radioactive, right? So in this set, it was the DNA that was radiolabeled. Therefore, this gave us proof that it is DNA of the virus that enters the bacterial cell and not the protein. So this confirmed that DNA is the genetic material. All right. Okay. Now, let's talk about some properties of genetic material. Now we know that DNA and RNA both can act as genetic material. In majority of organisms, DNA is the genetic material, and in some viruses, RNA also can be the genetic material. What are some properties that a molecule should have in order to act as a genetic material? Why can't proteins be the genetic material? Why can't lipids be the genetic material? What properties do they lack that DNA have, which makes DNA the genetic material? That's something we'll have to study. A molecule that has to act as the genetic material should fulfill a couple of criteria. There are 4 criteria. Firstly, it should be able to generate its replica, it should be able to make a copy of itself. Now, I have some information in my genetic material. If I should be able to transfer it to the next generation, to my children, I should be able to make a copy of itself and then pass it on. It's not like I will cut out like a little bit of information and pass it on to the next generation. Unless it's able to make a copy of itself, transmission of genetic information will not be possible. So if a molecule has to act like a genetic material, it should have the ability to make copies of itself, right? Next, it should be stable chemically and structurally. It's containing such important information, right? So it has to be extremely stable and not get changed. It should be stable both chemically as well as structurally, because it's containing like the most important information that a cell needs. Everything that's happening inside of a cell is under the control of DNA. So if there's any messing up that happens in its information, the cell will be affected. In turn, the organism also could be affected. So what molecule you have as a genetic material has to be very stable and less reactive, so that the information is protected very well. Okay. Third, it should provide the scope for slow changes that are required for evolution. So you know how we evolved from apes, right? Every organism, even right now, is undergoing evolution. So how does this evolution happen? How did an ape that was living on the trees become a human that is walking on its feet? How did we get this erect posture? How did we get these what do you call this, like we are able to hold this? Okay, in evolution, in zoology, you will learn. So all of these features our ancestors did not have, some of our ancestors did not have, and we have it. That happened because DNA, at the DNA level, there were some changes that took place, which we call mutation. So mutation will help in evolution. It should be stable. It shouldn't change a lot. It shouldn't change a lot, but some amount of change it should allow. Only then evolution can happen. Okay, so that's our third point. And finally, it should be able to express itself in the form of Mendelian characters. We saw how in the previous chapter, Principles of Inheritance and Variation, Mendelian characters were expressed from one generation to the next, right? We will not get into the details of it now. So a genetic material should have the ability to express itself in the form of Mendelian characters. So that's about the properties that a genetic material should have. Now we have DNA and RNA both as genetic materials. Do all do DNA and RNA follow all of those criteria? Do they fulfill all of those criteria? Let's understand. Firstly, it should be able to generate its copy. DNA, RNA both have the ability to generate their copies. How exactly it happens, I'll tell you later. Second, it should be stable chemically and structurally. When you compare the stability of DNA and RNA, definitely DNA is more stable compared to RNA. The stability of DNA is higher compared to RNA because of a couple of reasons. Firstly, because of if you look at the sugar ribose and deoxyribose, at the 2' carbon, you have a hydroxyl group in RNA, and here you have a hydrogen group. So this is relatively more reactive compared to this, so its stability is reduced. Apart from that, in DNA you have thymine, here you have uracil as a nitrogenous base. Compared to thymine, and when you're comparing thymine and uracil, thymine definitely is more stable than uracil. Apart from that, RNA also has catalytic activity. Sometimes it acts like a catalyst. So it has catalytic activity which will further reduce the stability of RNA. And whenever it is single stranded, it is usually RNA is single stranded and DNA is double stranded. Whenever it's single stranded, that will make it even more reactive and unstable. Because of all of these reasons, DNA is generally more stable compared to RNA. That is why DNA evolved from RNA. Okay. Now, it provides the scope for slow changes required for evolution. Definitely both of them provide, but it's more in RNA compared to DNA. Mutations rate is higher in RNA compared to DNA. Recently also when COVID happened, you were learning about how newer variants were coming every few months, right? That is because the coronavirus has an RNA genome. So that mutates at a much faster rate compared to our DNA genome, okay? Next, it should be able to express itself in the form of Mendelian characters, which both do. So that is check on both sides. All right. So relatively, because of DNA's stability over the course of evolution, that has been the preferred genetic material, and that is why that is the genetic material in most organisms. When life originated on Earth, the first genetic material that probably formed was RNA. Not probably, it is for sure the first genetic material that formed. RNA is essential to our life processes such as metabolism, translation, splicing, all of that involves RNA. What exactly these are, I'll tell you in a little while. RNA used to act like the genetic material as well as a catalyst. I told you it has some catalytic activity as well. But because it was a catalyst, it was very reactive and unstable. Therefore, DNA evolved from RNA with certain changes. Because DNA has double strands, it has a complementary strand, therefore it resists change compared to RNA. Therefore, DNA is the better genetic material because it is more stable. Okay? Okay. So far, we've been talking about the structure of DNA. How did we get to know DNA is a genetic material? What are the differences between DNA and RNA? What are some features a genetic material should have? All of that we've discussed. In the beginning of the session, I told you that DNA is the hero. He is the boss. It is the boss, rather. So, we say that everything is controlled by the DNA. But if you look at a eukaryotic cell, DNA is always sitting inside the nucleus. Only when the cell is dividing, the nuclear membrane disappears, it comes out as chromosomes, and immediately after cell division, it goes back into its cozy chamber, the nucleus. Despite sitting inside the nucleus, despite having such valuable information, priceless information, sitting inside the nucleus, how does it control everything that happens inside the cell? That can be explained by central dogma of molecular biology that was proposed by Francis Crick. This is the Francis Crick. This is the same person who proposed one of the people who proposed the double helical model of DNA. There I said Watson and Crick, right? So it's the same Crick who proposed the central dogma of molecular biology. So according to the central dogma of molecular biology, genetic information flows unidirectionally from DNA to RNA to protein. DNA itself will not do the job. It contains the information. It makes the protein do the job. DNA has the information we say all the time. It has very important, priceless, valuable information. How does this information, what exactly is this information? This information is in the form of a nucleotide sequence. That information basically helps in the production of proteins, and those proteins will do the job. So this happens in two steps. The first step is transcription, the second step is translation. DNA, being the genetic material, has the ability to make copies of itself by a process called replication. Now, let's take a eukaryotic cell. Here you have the nuclear membrane. Within the nuclear membrane, you have DNA. DNA has the information for the formation of a protein. But DNA doesn't come outside because the information is very, very valuable, very important. It cannot be messed up with. It should not be changed very frequently. So it's very protected and sitting there inside the nucleus. Now, the cell has to perform some job. And for that job, an enzyme is required. And that enzyme is a protein. Where will the protein form from? For the formation of that protein, information is in this segment of DNA, let's say. That information has to go to the ribosomes because proteins are synthesized in the ribosomes. Who will get this information in the DNA present inside the nucleus, take it to the ribosomes? There should be some molecule to do that. That is done by a molecule called mRNA. It's a type of RNA called messenger RNA. So what basically happens is, whichever segment has the information, from that segment, the information is copied. That information is brought out of the nucleus by a molecule called messenger RNA or mRNA. So it copies the message and gets it out. This mRNA next goes to the ribosome. Ribosome will read the message on this mRNA and it will synthesize a protein. The process by which the message in DNA is copied to RNA is called transcription. So this message copying is the first step, transcription. The message that is copied by RNA is in the form of a coded message. Remember, if you have to write secret letters to your friends, you use different things, right? For example, if somebody has to say I love you, they say 143. That is a coded message. So like that, whatever message DNA has, the DNA has it in the form of a coded message, and that coded message can be translated only by the ribosome. So this messenger RNA which has copied the information, it cannot read it. That message can be read only by the ribosome. For the translation of that message, it goes to the ribosome. The ribosome reads the message, and according to the message, it simultaneously synthesizes a protein. That protein goes on to do its job. The process by which the message in RNA is translated by the ribosome and simultaneously a protein is synthesized is called translation. That is the second step. So in the first step, DNA information is copied by RNA. RNA takes it, that is the transcription step. In the next step, the coded message has to be translated. That can be done only by the ribosome. So that RNA goes to the ribosome. Ribosome reads that message and synthesizes a protein. Whatever job the protein was supposed to do, that job will be performed by the protein. So different segments of DNA will have different information for different proteins. Depending on the cell's requirement, depending upon what the cell needs at that moment, different segments will be transcribed and translated, and the proteins will be formed. Protein will go on to do their jobs. That is how the DNA, despite being inside the nucleus, will control everything that happens inside of our cells. All right. So this is an overview of central dogma of molecular biology. We will learn in detail how transcription happens, how translation happens, and how replication happens, how transcription happens, and how translation happens. All right. So let's begin by talking about DNA replication. It's very clear that DNA is being replicated. That is why it's called DNA replication. Replica in general English means copy. You may have heard how people say you are a replica of your mother, or you are a replica of your aunt, or whoever. So it just means that it is the exact copy. So DNA replication is a process by which DNA will make an extra copy of itself. All right. So, this in eukaryotic cells will occur during the S phase of cell cycle. How exactly it happens? There were different people that proposed different theories. Some people said it happens by conservative mode, some people said it happens by semi-conservative mode, some people said it happens by dispersive mode. Now, what is the difference between these? Let's take a look. Here is the parent DNA molecule that is represented by blue strand. If this has to make an extra copy of itself, in the end you should have 4 strands, right? So, if by conservative mode, if it makes a copy of itself, both of these strands will go into one daughter cell. The other copy will have 2 newly synthesized strands. The newly synthesized strands are represented by red color. So, this is new. This is maternal or old. According to conservative mode, both the strands, parental strands, are conserved in one DNA and the other DNA molecule will get 2 brand new strands. According to semi-conservative, in both the DNA molecules that are formed, one strand will be one strand will be parental, the other one will be newly synthesized. This is semi-conservative. One strand is conserved, one is newly synthesized. In dispersive, it's like both the strands in both the DNA molecules will have a mixture of some old parts and some new parts. So people had different opinions about how replication takes place, but ultimately it was concluded that DNA replication happens semi-conservatively. So how exactly that happens? Let's take a look. This was proven by 2 people, Messelson and Stahl, in the year 1958. So what they basically did was, they took E. coli bacteria, they grew it in a nutrient medium, in this nutrient medium, they had added a heavier isotope of nitrogen. Normal nitrogen is N14. They added N15. Remember, N15 is not a radioactive isotope, it's just a heavier isotope that you can distinguish in density gradient centrifugation. So what they did was, they took these E. coli bacteria. What is a nutrient medium? The food that you give for bacteria to grow. If you're growing them in lab, you have to give them the food. So when bacteria grows, DNA replication has to happen, cell division has to happen, all of that will happen. So if DNA has to make an extra copy, it should make nucleotides, right? And within the nucleotides, you have nitrogenous bases. To make those nitrogenous bases, where will they get nitrogen from? From the nutrient medium. So whatever nitrogen is available in the nutrient medium, they will pick it up and make the nitrogenous bases and use the same bases to make nucleotides and eventually DNA. So they grew bacteria, E. coli bacteria, for several generations on a nutrient medium that contained the heavier isotope of nitrogen. And they confirmed that all of the bacteria in the colonies in that nutrient medium had N15 incorporated in their DNA. Okay, so they all had N15 incorporated in their DNA. E. coli bacteria divide once in every 20 minutes. Okay, so what happens here is, after 20 minutes, they transferred these bacteria to a culture medium that contained normal nitrogen N14. The bacteria, they extracted some bacteria and they spun it in a density gradient centrifugation. They performed a density gradient centrifugation. It's called cesium chloride density gradient centrifugation. So basically what happened here was, when you spin it at very high speeds, it will form bands at select specific regions. So when these bacteria were spun in the centrifuge, they got a single band at this position. Here, all of the bacteria had both strands that were that had the heavier isotope of nitrogen. So you have N15, N15 here. After 20 minutes, they took the bacteria again and they spun it. Again, they got a single band, but this time, this band's level was slightly above the previous one. So here, this indicates if it's slightly above means it is less dense. If it is less dense means it does not have both strands of N15. So that indicated that here was a hybrid strand. If it was by conservative mode, then you should have got two types of DNA, right? If it was conservative mode, we should have got two types of DNA. But because it is semi-conservative, all the DNA are of the same type and they form a single band which is slightly higher than this, indicating that it's little less dense. After 20 more minutes, they found out when they spun it in a centrifuge that there were two bands formed. One band here and the other band here. The lower band that was formed here was identical in position to this band, and the new band that formed was slightly above it. When would this happen? Whenever there are two types of DNA. Because there are two types of DNA here, first type which have both newly synthesized N14 strands and the second type which are hybrids. Because there are two types of DNA, you have two bands formed, and one of them is the hybrid band, the other one is both N14 band. This could have happened only if DNA was semi-conservative. When will you get a slightly higher band of hybrid? Whenever DNA is semi-conservative in replication. Only when both of these strands are present in the DNA molecules and one newly synthesized strand is formed, only then can you get this kind of band. Now what happens when you transfer this to a medium containing N14? For whatever new strands have to be synthesized, they will have to make use of that N14. N15 is no longer available. So for the strands that are newly synthesized, they would have incorporated this N14. So the new strands will be lighter, the old initial strands will be heavy. What happens after the next round of replication? Both of these will act like parents here. So over here, this is the parent from this and this is the parent. At this point, you don't have N15 in the medium anymore. So whatever N14 is available, from that the new strand will be synthesized. Same thing happens for this. As a result, at the end of this, you will get two hybrids and two N14, purely N14 DNA molecules. So this indicated that replication of DNA happens by semi-conservative mode. Okay. Now, another person called Taylor, he also performed similar experiments using radioactive thymidine on a plant called Vicia faba. It's also known as faba beans. This also helped in concluding that replication of DNA happens semi-conservatively. Now let's understand how exactly this process of DNA replication takes place. Alright, so when DNA replication happens, you start with one molecule of DNA that has two strands, right? So you'll have two strands. After replication, an extra copy is made. We know that it happens semi-conservatively. So one of the parent strands is retained in both the daughter DNA molecules and a newly synthesized strand forms, right? So this is basically what happens during replication. So for replication to take place, the information in DNA has to be copied. And this happens based on the principle of complementarity. What does that mean? We already know that adenine forms 2 hydrogen bonds with thymine and guanine forms 3 hydrogen bonds with cytosine, right? So this is called complementary base pairing. So based on this, two newly formed DNA strands will get synthesized. So how exactly that happens? Let's take, for example, a strand of DNA that has the sequence AATGCG on one strand. On the other strand, you'll have TTACGC. Okay, so here you'll have two double bonds, and over here you'll have three hydrogen bonds. So when DNA replication has to happen, the first thing that happens is the hydrogen bonds will break, the two strands will separate, and you will have two separate strands that have AATGCG, and over here you'll have TTACGC. There are enzymes called DNA-dependent DNA polymerases that are capable of reading what is given in one strand, the nucleotides or the nitrogenous bases that are present on one strand, and add complementary nitrogenous bases or nucleotides to it. Okay, so this DNA polymerase goes on reading the nucleotide sequence here, and when it reads A here, it will add a complementary T. When it reads A here, it will add T. When there is T in one strand, it will add A. So like that, it will go on forming another strand based on the principle of complementarity. It will go on adding nucleotides with complementary bases. Same goes for the other strand. So the DNA polymerase reads that there's a T in the template strand, it will add an A. It sees there's a T, it will add an A. If there is an A, it will add a T. If there is a C, it will add a G. If there is G, that will add a C. So likewise, after the process of replication is completed, you will get two copies or two molecules of DNA from one copy, and how this happens is based on the principle of complementarity. For this to happen, the bonds have to break. For the bonds, hydrogen bonds in the nitrogenous base pairs to break, you need enzymes. And to add new nucleotides complementary to the template strand, we need enzymes. Okay, a lot of enzymes are involved in the process of DNA replication. We learn about them shortly. First thing we have to remember, one thing about DNA replication, it is DNA replication cannot begin anywhere in the DNA molecule. There is a specific sequence of nucleotides at which replication will start. That is called the origin of replication. So that is called the origin of replication. It's also called as the ORI site. In eukaryotic cells, replication happens in the S phase of the cell cycle. In prokaryotes, you usually have one origin of replication. Since eukaryotic DNA is much longer than prokaryotic DNA, they will have multiple origins of replication. So what basically happens in origin of replication? If you take this as a segment of DNA, and let's say this is the region that contains the origin of replication. The sequence of nucleotides here is the origin of replication. So replication ideally should start here. So what happens there is, the hydrogen bonds that exist between the nitrogenous bases will break, and a bubble sort of thing forms, as you can see here. Over here, there is no pairing. The hydrogen bonds have been broken. This is the origin of replication. So over here, a bubble kind of structure is formed, which we call the replication bubble. Replication proceeds on both sides of the replication bubble. So from this point onwards, the enzyme will start reading the nucleotides here and move this way. Replication will proceed on the opposite direction as well. So what exactly happens on one side is what we will learn. In a replication bubble, you have two forks. So this half of it is called one replication fork, and this half is called the other replication fork. So what you're looking at here in this NCRT diagram is one half of the replication bubble. Wherever there is the replication bubble, you will have two forks. What you're looking at here is one fork. Over here, replication proceeds in this direction. Okay. So Over here in the fork, wherever the region it is forked, over there the hydrogen bonds are broken. For the breaking of hydrogen bonds, we need an enzyme called helicase. Okay, wait. Okay, we need an enzyme called helicase. This helicase breaks hydrogen bonds. What happens next? Based on the principle of complementarity, I said there is an enzyme that will add nucleotides complementary to the template strand. Now, what is a template strand? These two strands which are present in the parent DNA molecule, those are called the template strands. Based on the information present in the template strand, based on the nucleotide sequence present in the template strand, complementary nucleotides are going to get added on the strand that is going to be synthesized newly. For that, we need an enzyme that is called DNA dependent DNA polymerase. So what does this DNA dependent DNA polymerase do? It polymerizes DNA. We already know that DNA is a polymer of nucleotides. When I say it polymerizes DNA, I mean to say that it goes on to add more and more nucleotides and make this chain extend. What exactly will be the sequence on this? Depends on the sequence in the template. If in the template there is an A, this enzyme DNA dependent DNA polymerase will add a T. If there is a G here, it will add a C here. So likewise, it will go on continuing to add nucleotides in the newly synthesized strand. Over here, we must understand one thing. This DNA dependent DNA polymerase is called so because this enzyme absolutely requires a template to produce a new strand. Without a template, it cannot synthesize a new molecule or a new strand of DNA. Because it depends on a template to synthesize a new strand, we say that it is DNA dependent. Why we call it DNA polymerase is very obvious, because it's polymerizing DNA. That's why we call it DNA polymerase. So this DNA dependent DNA polymerase works only in the 5' to 3' direction. It can add new nucleotides only in 5' to 3' direction. But in the parent DNA molecule, you have two template strands, one which runs from 3' to 5' and the other which runs from 5' to 3'. That is because DNA is anti-parallel, right? So what happens here is, because DNA dependent DNA polymerase can only function and polymerize DNA in 5' to 3' direction, you can see how in one of the strands, the continuous synthesis is seen. DNA will get continuously synthesized. Because it can work only in 5' to 3' direction on another strand, on the template strand which has the polarity 3' to 5', it will get continuously synthesized towards the fork. On the template strand that has the polarity 5' to 3', it will get synthesized for sure, but it will get synthesized discontinuously away from the fork. Wherever continuous synthesis happens, that is called the leading strand. Wherever discontinuous synthesis occurs, that is called the lagging strand. Okay, so whatever is happening over here is going to happen over on the other side as well. So in one side of the fork, if this is the leading strand and this is the lagging strand, on the other side of the fork, this will be the lagging strand and this would be the leading strand. So replication is going to proceed in both directions. The helicase enzyme is going to break hydrogen bonds this way as well as this way. DNA polymerase is going to add new nucleotides complementary to the template strand in both directions. So at the end of replication, you will have 4 strands of DNA or 2 molecules of DNA. Alright, so that's about leading and lagging strand. Another thing that we must understand here is something is being synthesized, an extra molecule of DNA or an extra copy of DNA is being synthesized. I've told you multiple times even before that whenever something is getting synthesized inside of a cell, energy is required. DNA replication is a very energetically expensive task for the cell. It consumes a lot of energy. And that energy is provided by the nucleotides themselves. Now, if you look at um, DNA dependent DNA polymerase, I said it will read the sequence of nucleotides in the template strand and add complementary nucleotides. Over here, the nucleotides have two functions. It has dual functions. One thing, the nucleotides themselves will provide the energy required for this polymerization while simultaneously acting like the substrates for DNA polymerase. You know how ATP provides energy, right? ATP is adenosine triphosphate. What is it? It is nothing but a nucleotide. Where is the energy present in ATP molecule? It's present in these two bonds. They are high energy bonds. Whenever those bonds are broken, energy is released. So what happens is DNA polymerase will bind to ATP. It could be ATP, TTP, GTP, CTP, any of these. Okay, so they are all nucleotide triphosphates that have a lot of energy in the bonds. So, if this DNA dependent DNA polymerase has to synthesize a new strand, it will read that it has an A there in the template. So, complementary to it, it will add a T. This T will be TTP, triphosphate. While this is forming hydrogen bonds with A, these bonds will be broken. So, there is pairing happening between these two. The energy that is released during the breaking of these bonds will be used in polymerization. Similarly, if you see T here, there will be A here. This A will be an ATP. They will be deoxy nucleotide triphosphates. So they will have energy-rich bonds in them. While being added during the polymerization of the new strand of DNA, the same energy is going to be used. All right. So T binds with A, C binds with G, G binds with C, and so on. So over here, we must remember that the nucleotides that are acting like substrates during the synthesis of a new strand of DNA will also provide the energy that is required for the synthesis of the new strand. All right. So another thing is this DNA dependent DNA polymerase does not know where to start. In order to give it a starting point, there is another enzyme called RNA primase that is required. The name of this enzyme is not mentioned in NCERT, so you need not bother much about it. Just know that DNA dependent DNA polymerase cannot by itself start the process of replication. It needs another enzyme. And wherever discontinuous synthesis has happened, there you have DNA segments produced in short fragments. It's not continuous. That's why it's called discontinuous. These are called Okazaki fragments. Later, these are joined by an enzyme called DNA ligase. What is ligation in general English? Ligation means joining. So this DNA ligase enzyme joins the fragments of DNA together. So that's about DNA replication. It is a very quick process. For example, if you consider prokaryotes like E. coli, apparently every minute 2000 nucleotides get added. That's how quickly replication proceeds. That's how efficient DNA polymerase is. And also they divide once in every 20 minutes. So this replication process pretty much takes about 18 minutes in E. coli bacteria, and it happens very quickly and very efficiently. So yeah, that's about DNA replication. Now let's move on and learn about transcription. I've already told you what transcription is. Transcription is the process where the information in a region or a segment of DNA is copied onto a strand of RNA. So, there are a couple of differences between replication and transcription. In replication, the entire DNA gets copied. Whenever replication takes place during the S phase, the entire DNA molecule will get copied and an extra copy is made. During transcription, only specific regions of DNA which contain information for the synthesis of a protein that the cell requires at that particular time. Only that will get transcribed. Secondly, in DNA replication, both the strands are used as the template and two new strands of DNA are synthesized. In transcription, only one of the two strands of DNA acts like the template, and from that we are going to get only one strand of RNA. Okay, so excuse me, if you were to define transcription, we can say that it is the process of copying genetic information from one strand of DNA. Only one of the two strands is acting like the template. From one strand of DNA to RNA, you can copy, the genetic information is copied. So how exactly does this happen? What is the principle that governs this? It is the exact same principle behind DNA replication, which is the principle of complementarity. So the principle of complementarity is what is involved in the process of transcription as well. The only difference is over here, DNA is the template, RNA is being synthesized. DNA has T, doesn't have U. RNA doesn't have T, has U. In DNA, you have thymine and not uracil. In RNA, you have uracil but not thymine. That is the only difference. Alright. So, during transcription, if DNA has A, T, C, G in its sequence, complementary to A, if you were synthesizing RNA, you do not have T. So, in place of T, you will add U instead. Complementary to T, A will be the nucleotide that will get polymerized. Complementary to C, you have G. Complementary to G, you have C. So, the only difference is that adenosine forms a base pair with uracil instead of thymine because RNA does not have thymine. Like I already mentioned, unlike replication and transcription, there's only a segment of DNA that will get transcribed and only one of the strands is used as the template. So, you know that DNA has two strands. One strand runs from 3' to 5' direction. The other strand runs from 5' to 3' direction. I'm repeatedly saying that only one of these two strands is going to act as the template. The strand that is going to act as the template will have the polarity 3' to 5'. Okay. The reason why both of these strands cannot act like the template is because, number one, if both of them act as the template, let's take a look at the sequence. That will make it clearer for you. A, G, C, T, T, A, G, C, T, C, G, A, T, C, G. So this is the sequence. If you compare the sequence of both these strands from 3' to 5', this is already in 3' to 5'. This, if you take it from 3' to 5', you will have the sequence 3'. It's the exact same sequence. I'm just reversing it. You'll have 3', G, C, T, A, G, C, T. Okay. So this and this will act as the template because for a strand to act as the template, it has to have the polarity 3' to 5'. Similar to DNA-dependent DNA polymerase, which could polymerize only in 5' to 3' direction, the enzyme involved in transcription also can work only in 5' to 3' direction. If it can polymerize only in 5' to 3' direction, the template should obviously have the opposite polarity, which is 3' to 5'. So, basically how proteins are synthesized is determined by the sequence of nucleotides. You know that nucleotides join to make DNA. From that DNA, you get nucleotides RNA by transcription. The sequence of nucleotides here will determine the sequence of amino acids in the protein. So what is basically going to happen during translation? Protein synthesis is going to take place. How do proteins differ from each other? Because of the sequence of amino acids in them. So what determines the sequence of amino acids in a protein? the sequence of RNA. So, every 3 nucleotides in RNA will have information for one amino acid. The next 3 nucleotides in RNA will have the information for the next amino acid. So, whatever is the sequence of RNA here, that will determine the sequence of amino acids in your proteins. So, if you look at this strand and this strand, you can see how the sequence is not the same. From the same segment of DNA, if you consider them both in 3' to 5' direction, which is the direction in which transcription proceeds, you can see how the same segment will code for 2 different proteins. That will complicate the entire mechanism of transmission of genetic information. So, in order to not have that confusion, avoid that confusion, only one of the 2 strands will act as the template. Another issue that would arise if both of them are used as template is, whenever a complementary RNA is formed to this strand, both of these will be complementary to each other and they will end up forming a double bond. That RNA will become double-stranded. Double-stranded RNA cannot be translated. Ribosomes cannot read the message in the double-stranded RNA. Also, our cells have enzymes that will assume any double-stranded RNA is viral RNA or bad RNA and break it down. So, in that way, translation will not happen, protein synthesis itself will not happen. Because of these reasons, both the strands in DNA cannot be used as templates for transcription. Alright? Now, what is a transcription unit? Like I already mentioned, there's only one segment, a certain segment of DNA that will get transcribed. Whichever segment of DNA gets transcribed, that is called the transcription unit. Now, a transcription unit typically has 3 parts or 3 components. There is the promoter, there is the structural gene, and there is the terminator. The promoter acts as the starting site for transcription. The terminator, as its name suggests, acts as the stopping site or the ending site for transcription. What lies in between is the structural gene. This is double-stranded DNA. So, only one of them will be used as the template. Whichever strand has the polarity 3' to 5', that is called the template strand. The strand that has the polarity 5' to 3' is called the coding strand. You might be confused or surprised why this is called the coding strand even though it is not involved in transcription. Let's take a nucleotide sequence for example so that you can understand easily. So here we have 3', 5', 5', 3'. This is DNA. Whatever is in orange is DNA. We'll have the sequence A, T, C, G, C, G, T, A, T. Here you'll have T, A, G, C, G, C, A, T, A. This is acting as the template. So, complementary to this, a new strand is going to get synthesized. And that new strand will be of RNA. It will be 5' to 3' polarity it will have because the template is 3 to 5. This will be 5 to 3. Complementary to A, it cannot add a T. So, instead it will add a U nucleotide. Complementary to T, it can add an A. You will have G, C, G, C, A, U, A. Now, if you compare the sequence of the template strand, the coding strand, both of which are strands that you find in DNA, with the newly formed RNA strand. Compare template and coding strand, they are complementary. Compare template and RNA, they are complementary. If you compare template with the coding strand, you can see how they are very, very similar in most places except for wherever you have T in DNA coding strand, you will have U in RNA strand. Except for this one difference, the RNA sequence is very similar to the sequence of the coding strand. Because it is RNA that is ultimately carrying the information to the ribosome, and coding strand has a similar sequence, we call this the coding strand, though it is not involved in transcription. The strand that is involved in transcription, the template strand, will have a complementary nucleotide sequence compared to the RNA that is newly synthesized. Alright, so that's about the different parts of the transcription unit. Now let's understand how exactly the process of transcription takes place. So for transcription to take place, we need an enzyme. Unlike in DNA replication where we had ligase and helicase and all of these enzymes, over here, breaking of hydrogen bonds and synthesis of new strand of RNA, all of that is taken care of by one enzyme itself, and that is called DNA dependent, bad choice of color. Yeah. So it's called DNA dependent RNA polymerase. Over here, the template is DNA, so it's dependent on DNA. The enzyme is RNA polymerase because RNA is being polymerized, RNA is being synthesized. This DNA dependent DNA polymerase knows to read the sequence of DNA nucleotides in the coding, the template strand, and add complementary nucleotides of RNA. But it does not know where to start. It does not know how to identify the starting site promoter. To help in identification of the promoter region, this DNA dependent RNA polymerase will transiently bind with a protein factor known as the sigma factor. This sigma factor will associate with this enzyme and help it to identify the promoter region. That is when the process of transcription is initiated. What happens after that is, once the promoter region is identified, DNA dependent RNA polymerase will go on to read the nucleotides in the template and add complementary RNA nucleotides and synthesize the RNA strand. Again, as it proceeds, it does not know how to identify the terminator, it does not know where to stop. In order for the RNA polymerase to stop at the terminator, it needs the help of another protein that is called the Rho factor. So this Rho factor will help in termination. So the first step, initiation, is when RNA polymerase binds to the sigma factor, identifies the promoter. After that, it will elongate the new strand of RNA based on the principle of complementarity. It associates with Rho factor towards the end of elongation. Rho factor helps in the identification of the terminator sequence, and that ends the process of transcription. So at the end of transcription, you will have a newly synthesized strand of RNA that is complementary to the template strand and has the sequence similar to the coding strand. Okay, excuse me. So, over here, we know that DNA acted like the template. One of the strands of DNA acted like the template, and it helped in the synthesis of a strand of RNA, which we call the messenger RNA. When we define a gene, we've learned about gene in the previous chapter as well. Gene is considered to be the functional unit of inheritance. It is considered to be the functional unit of inheritance, and any segment of DNA that codes for or has the information for any type of RNA. It could be tRNA, rRNA, mRNA. Whichever segment of DNA has the information for the synthesis of an RNA is called a gene. What is a cistron then? A cistron is basically a type of gene that specifically codes for mRNA, and via the mRNA, it codes for a polypeptide. A segment of DNA that specifically has the information to code for a polypeptide or a protein is what we call a cistron. In prokaryotes, generally in one gene, you will have information for more than one polypeptide. Such genes are said to be polycistronic. In most eukaryotes, one segment will have information only for one polypeptide synthesis. Such genes are said to be monocistronic. So that's the difference between a gene and a cistron. A cistron is basically a type of a gene that specifically codes for mRNA, which has information for the synthesis of a polypeptide. All right, so that's important for you to know. Over here, in with respect to this enzyme, DNA-dependent RNA polymerase, in prokaryotes, the DNA-dependent RNA polymerase, it's of just one type. That single type of RNA polymerase itself is capable of transcribing all types of RNA, including mRNA, tRNA, rRNA, and all the different types of RNA. But when it comes to eukaryotes, we have at least 3 different types of RNA polymerases that we find in the nucleus. Transcription occurs in the nucleus. Replication also occurs in the nucleus in eukaryotes. So, we have at least 3 different types that are involved in the synthesis of different types of RNA molecules. RNA polymerase 1 is involved in the synthesis of ribosomal RNA, specifically 28S type, 18S type, and 5.8S type. RNA polymerase 2, that is responsible for the synthesis of hnRNA. hnRNA is short for heterogeneous nuclear RNA. This basically is a precursor. It's a precursor of mRNA. We'll learn about this in just a little bit. And we have RNA polymerase 3 that is involved in the transcription of tRNA, 5srRNA, and small nuclear RNAs. So it's important for you to know this. Okay. Apart from that, are there any differences between prokaryotes and eukaryotes when it comes to transcription? Yes, there is. Firstly, transcription in prokaryotes, it will happen in the cytoplasm itself because there is no membrane-bound nucleus. But in eukaryotes, it happens within the nucleus. Okay. One thing that we must understand about eukaryotic genes is that in eukaryotes, you have split gene arrangement. What does that mean? I already said that our DNA is 6.6 into 10 to the power of 9 base pairs long. It's huge. Their entire DNA length will not carry information for the synthesis of proteins. There are certain regions where there is information for protein synthesis, and there are certain regions which lack information for protein synthesis. We discussed this when we spoke about heterochromatin and euchromatin, right? So, I already also mentioned that in eukaryotes, the genes are monocistronic. So, you have one promoter, you have a terminator, and over here, this segment is the structural gene. This structural gene will in most cases have the information for the synthesis of one polypeptide, which is why we call it monocistronic. Even within the structural gene, there are certain regions that have information and there are certain regions where there is no information for the synthesis of polypeptide. The regions which do have information and express themselves are called exons. The intervening sequences which do not code for any polypeptide, which lack information, these are called introns. since the regions that contain information or the exons are split and are interspersed by introns, we say that the genes in eukaryotes are split genes. Now, when transcription happens within the nucleus, RNA polymerase cannot distinguish whether uh, it is an intron region, it is an exon region, and transcribe accordingly. All it does is whatever is present between the promoter and terminator, it goes on transcribing the entire sequence. As a result, the mRNA that we get will have regions that have information and regions that do not have information. Introns which do not have information are called non-coding sequences. The exons that do have information are called the coding sequences. So before you send out this information to the ribosomes to be translated, you have to remove all the unnecessary regions, intron regions, and join together the exons. So that is something that has to happen in the nucleus before the RNA goes out to the ribosome. This RNA that is synthesized, which has both introns and exons, this is called hnRNA. The RNA that forms as a result of this, which will contain both introns as well as exons, such an RNA is called hnRNA or heterogeneous nuclear RNA. Heterogeneous because it has introns and exons, nuclear because it was synthesized in the nucleus. So this is called heterogeneous nuclear RNA. This is exactly what we were talking about over here. Okay, so that's transcribed by RNA polymerase 2. So this heterogeneous nuclear RNA has to undergo some modifications before it goes to the ribosome. One modification for sure is that these non-coding sequences have to be removed and the coding sequences have to be joined together. Apart from that, two other things will take place. Okay, so if the RNA, single-stranded RNA that has been newly transcribed, goes from the nucleus out to the cytoplasm through the nuclear pore, there will be enzymes in the cytoplasm that are capable of breaking this down. In order to protect itself at the ends, the enzymes are called exonucleases, and they are capable of breaking this RNA only from the ends, either 5' end or 3' end. So before this goes out to the nucleus, you have to protect its ends. In order for that, two processes will take place. Firstly, capping will occur. It's called G capping, where a modified G, methyl guanosine, is added to the 5' end of the newly synthesized hnRNA. That process is called capping. This protects this end from the activity of exonucleases. The second step is called polyadenylation, which occurs at the 3' end. So what happens at 3' end is, as many as 200 to 300 A's are added in a template independent manner. What does that mean? For this RNA to be synthesized, DNA template was required. But for this poly A tail to be added, no template is required. This also protects the 3' end of the RNA. The third step is splicing. In splicing, basically what happens is the introns get removed, the exons are joined together. As a result, whatever you get, that is the functional mRNA. This is what will go to the ribosome to get translated. Okay, so these are some post-transcriptional modifications that take place in eukaryotes. Now let's talk about the difference between prokaryotic transcription and eukaryotic transcription. Number one, in prokaryotes it happens in the cytoplasm. In eukaryotes it occurs in the nucleus. Second point, there's just one type of RNA polymerase that's capable of transcribing all the different types of RNAs in prokaryotes. When it comes to eukaryotes, we have at least 3 different types of RNA polymerases. The RNA that is formed as a result of prokaryotic transcription is polycistronic, meaning the same segment will have information for more than one polypeptide. Whereas in eukaryotes, it's monocistronic. It will have only information for one polypeptide that also is split. Fourth, Yeah, this is a repeated point, so ignore this. Next. Or you can just say that there is no post-transcriptional modification required in prokaryotes, but in eukaryotes, post-transcriptional modifications like capping, tailing and splicing, all of that will take place. Okay. Okay. So, we know now the DNA has information. This information is in the form of a nucleotide sequence. This information is copied to a strand of RNA, mRNA specifically, and that has to go to the ribosome. And in the ribosome, it reads this message and simultaneously synthesizes a polypeptide. So, how exactly does the ribosome read the message and help in the synthesis of polypeptide? What exactly is the code here? We've been saying that it is the information is in the form of a code. It is in a coded format that can be decoded or deciphered only by the ribosome. We've been saying this all along. So, what exactly is this code? This is called the genetic code. There is this one person called George Gamow. He believed that the code should constitute a combination of bases. Why, you ask? In DNA or even RNA, you just have 4 types of nucleotides. In proteins are made up of 20 different amino acids. If each nucleotide coded for 1 amino acid, we would have enough codes only for 4 amino acids. What about the remaining 16 amino acids? It's not possible for one nucleotide to code for 1 amino acid. Therefore, obviously, it should be a combination of bases. If it were a combination of 2 bases, that would be 4 nucleotides to the power of 2. It would give us 16. But the amino acids are much greater, 4 more. So, it probably is not a combination of 2 bases. But if it's a combination of 3 bases, you get a total of 64 combinations, which we call the codons. This is much higher than the number of amino acids that are involved in protein synthesis. So, he assumed that the code should be made up of at least 3 nucleotides. That would generate 64 codons. For his hypothesis, many people provided proof. Some of them were our Indian scientist Hargobind Khorana. We also have Marshall Nirenberg. So what Hargobind Khorana did was, he synthesized RNA molecules with his choice of combination of bases. He artificially synthesized RNA in the lab, and he found out what are the different amino acids that you could get from such a sequence. Marshall Nirenberg, he developed a cell-free system for protein synthesis. So usually protein synthesis happens inside of our cells, inside of our ribosomes, but he developed a method where you could synthesize proteins without ribosomes. And also there is an enzyme called Severo-Ochoa enzyme. This enzyme is helpful in polymerizing RNA with a defined sequence in a template independent manner. That means even without DNA, with the help of this enzyme, they could synthesize RNA. So they synthesized RNA sequences with their choice of nucleotides. They could synthesize proteins in cell-free systems. They knew what their RNA sequence was. They identified what was the amino acid sequence in the proteins. As a result, they deciphered the genetic code. They found out which 3 nucleotides exactly coded for which amino acid. And as a result, we have this checkerboard. Okay. So here you can see how every 3 nucleotides of RNA or every 3 bases will code for a particular amino acid. For example, over here you have AUG. AUG codes for methionine. Here everywhere you see U and not T because this is about the RNA, transcribed RNA that will have the information. There are certain codons that will code for there are certain amino acids which will have only one codon. There are some amino acids which will have more than one codon. So basically, you can see how there are 64 codons that are coding for 20 amino acids. Of these 64 codons, 3 codons do not code for anything. They are called the stop codons. Okay. So let's learn about some features of the genetic code. Firstly, it is clear now that the codon is triplet. Out of 64 codons, 61 code for amino acids and 3 codons do not code for anything. Which are those 3 codons? UAA, UAG, UGA. One easy way to remember this is that UAA is you are away, UAG is you are gone, UGA is you go away. Okay, so that's one way to remember these 3 codons. There are some amino acids that are coded by more than one codon. That property is called degeneracy or degenerate. Here you can see clearly how leucine is coded for by 6 codons. 1, 2, 3, 4, 5, 6. All of these codons code for leucine. Leucine can be coded for by more than one codon, but these codons will always code for leucine itself. There is no ambiguity there. So it's called a non-ambiguous code or an unambiguous code. Leucine can be coded for by more than one codon, that is degeneracy. UAA will always code for leucine, not phenylalanine, not serine, not tyrosine. Always for leucine. There's no confusion for these codons. That property is called unambiguous nature, or basically they don't have any confusion in what amino acid they have to code for. Okay, so that's one property. The codon is read in a continuous fashion. What does that mean? If this is an RNA strand that has A, U, G, C, U, G, C, U, A, the codon is read in, the information is read in units of 3. Okay, so this will be one codon. The next codon cannot be like this where C is skipped. It is read continuously. So, the next codon will be this. The codon after that will be this. No codons are skipped in between, or no nucleotides are skipped in between. That is what it means when we say it is read in a continuous fashion. The code is nearly universal. What does that mean? If UUU codes for phenylalanine in me, it codes for phenylalanine in you, in a mouse, in an ant, in a plant, in a bacterium, everywhere the codon are pretty much the same. Why we are saying nearly universal is because there are some exceptions to this, such as certain mitochondrial codons and codons in some protozoans. Apart from that, pretty much in every organism, it is the same. Fifth point is that AUG has dual functions. I told you which ones are the stop codons, but I didn't mention which is the start codon. The start codon is AUG. One function of AUG is that it codes for the amino acid methionine. Another function is that it acts as an initiator codon. Just like we had promoter for transcription, you have initiator codon and terminator codon for translation. In any protein synthesis, the mRNA at the 5' end, the first codon is going to be AUG. That's why it's called the initiator codon. That is one function that it performs. It acts as the initiator codon. Also, it codes for an amino acid, methionine. Okay? So, that's about some features of the genetic code. Now, let's talk about mutations. Mutations are of so many different kinds. Point mutations are mutations that happen at one point at one nucleotide in DNA. It could be a substitution type of mutation where instead of A, T is G, C, G, instead of this you have A, G, C, G, C. T is replaced by A. So replacement, I'm just showing it in one strand, it will happen in the opposite strand as well. So that is called a substitution mutation. Insertion is when to this one or more nucleotides are added. A, T, T, C, G, C. Over here at this point, one nucleotide is added. Deletion is when one or more nucleotide or base pairs are removed. So it will be A, C, G, C, where T is removed. I'm showing it only on one strand, but that will happen on both strands. That's why we're seeing base pair. Whenever there is an insertion or deletion, there will be a shift in the reading frame, and therefore it is also called frame shift mutation. Before mutation, whatever will be the reading frame, during translation, that will be altered. From the point of mutation onwards, the codons will no longer be the same. The sequence of nucleotides is going to change. As a consequence, the codons and the sequence of amino acids is also going to change. Okay, so that's about these point mutations. Now let's talk about the different types of RNA. In translation, many types of RNA are involved. Let's learn about them. The first type is called mRNA. mRNA is short for messenger RNA and it provides the template like I already said. tRNA brings amino acids and reads the genetic code. How exactly it does that, we'll learn about it later. It's short for transfer RNA. We have rRNA. rRNA is a structural component of ribosomes. I've been saying since the start that protein synthesis happens in ribosomes. What are ribosomes made up of? Ribosomes are made up of RNA and about 80 different types of proteins. The RNA component of ribosomes is what we call ribosomal RNA. It is structurally a component of ribosome, so it has a structural role as well as a catalytic role. RNA very rarely acts like a catalyst. In most cases, proteins are the enzymes. RNA acts like the catalyst during formation of proteins when two amino acids have to be linked together by peptide bonds. That bond formation is catalyzed by RNA, okay, ribosomal RNA. That is its catalytic role. Okay, so we know about mRNA already. I already told you what mRNA is. We know that RNA, ribosomal RNA is a component of ribosomes. Let's study about tRNA, transfer RNA. Now, if you look at mRNA that is carrying the message, in mRNA, the message is in the form of nucleotide sequence. Every 3 nucleotides here will carry the information for one amino acid. Okay, so in a protein, you will have amino acid. In RNA, you have nucleotides. The nucleotides can form complementary bases with nucleotides but not with amino acids. There should be a molecule that can form base pairs with this and accordingly bring the corresponding amino acid and basically act like an adapter. That job is performed by our tRNA molecule, which is why it is also called the adapter molecule. Similar to how in mRNA you have these codons, in tRNA you have anticodons. Let me give an example. Let's say this is an mRNA. Let's take AUG. We already know AUG codes for methionine. AUG is the codon that you have in mRNA. Complementary to this, in tRNA, you will have UAC. This tRNA which has UAC as the anticodon, on the other end will have the amino acid methionine. So this will help in the synthesis of proteins by bringing the corresponding amino acids inside the ribosome for protein synthesis. So this will on one end read the codon, it will have the complementary anticodon. Corresponding amino acid it will carry on the opposite end. That is how it acts like an adapter. So it has an anticodon loop that has bases complementary to the codon in mRNA. On the other end, it has an amino acid acceptor end to which it binds to the amino acids. There are specific tRNAs for each amino acid. There is a specific tRNA for initiation, which is called the initiator tRNA. For the stop codons UAA, UAG and UGA, there are no stop codons. There are no tRNAs. If you look at its secondary structure, it looks like a clover leaf. You know what a clover leaf is, right? In this game of cards, you have this. This is called a clover leaf. So, in its two-dimensional structure, it looks like that. But in three-dimensional structure or tertiary structure, it looks like an inverted L. You can see how here the codon reads AGU. AGU codes for serine. So this tRNA will have an anticodon UCA complementary to this codon, and it will bring serine inside the ribosome for protein synthesis. Okay, so that's about tRNA molecule. In its tertiary structure, it looks like an inverted L. All right. Now, let's talk about how exactly translation takes place. Translation is basically the process by which amino acids are joining together, polymerizing and forming a polypeptide. Where does this happen? It happens inside the ribosomes. How does this happen? Let's learn that. For initiation of translation, what happens is you have the mRNA. This mRNA first binds to the small subunit of the ribosome. You have two subunits in the ribosome, small and large, right? So at the start codon, at the 5' end, the small subunit will bind to the ribosome. Over here also protein synthesis, something is being synthesized, it requires energy. So before the translation is initiated, tRNAs don't always have amino acids attached to their end. Before the process of translation, amino acids will get activated by making use of ATP, and those activated amino acids will bind to the amino acid acceptor end of tRNA by a process called amino acylation or charging of tRNA. Then what happens is the initiator tRNA that carries UAC as the anticodon on one end and methionine on the other end will go and bind to the initiator codon. Okay. Then the large subunit will come and bind to it. Within the ribosome, there's enough space for two tRNAs to fit. So the first tRNA will come and bind to one side, corresponding to the codon. In the next side, the next amino acid will come and bind. The next tRNA will come and bind. When two tRNAs are brought close to each other, formation of peptide bond between them is favored. Normally, it will happen when two tRNAs are brought together also. But within the ribosome, in the large subunit, in prokaryotes, you have enzymatic activity. There is RNA acting like an enzyme called ribozyme. This will catalyze the bond formation between these two. Once a peptide bond is formed between these two amino acids, the tRNA here will leave. Now this ribosome will move, translocate to the next codon. So whatever tRNA is here will move to this position and the second position will be vacant. Corresponding to the next codon, the next tRNA will get into the ribosome. It will form base pairs between codon and anticodon. When these two tRNAs are brought together, the amino acids will form peptide bond. After that, this will move out. Ribosome will slide over mRNA. The next site is vacant again for the next tRNA. In that way, it will go on elongating. Once it reaches the stop codon, remember for stop codons, we do not have any tRNAs. Once it reaches the stop codon, there is a protein factor called a release factor that will bind to the stop codon and terminate the process of translation. At the end of it, we will have our new strand of polypeptide and the RNA would have done its job. Okay, so that's how translation takes place. Now let's learn about the regulation of gene expression. You know that our cells have a lot of DNA, and in many segments of DNA there is information for the synthesis of polypeptides. But it's not necessary that we need all the polypeptides all the time. There are some enzymes, certain proteins that are constantly required by the cells, and there are certain enzymes or proteins that are not required at all times and are required only now and then based on the requirement of the cell. How does the cell know when to turn off that gene, when to turn on that gene? Whenever there is a requirement that arises, how will the cell know to turn on a particular gene and produce its product? And whenever there is no requirement, how does the cell know to turn it off? How is the expression of genes basically regulated? It can be regulated at various levels. In eukaryotes, it can happen at transcription level, where transcription itself is not happening. If transcription is not happening, RNA is not produced, so obviously protein will not be synthesized. You know, after transcription, post-transcriptional modifications of capping, tailing and splicing will happen. If that does not happen, then translation will not happen. mRNA is formed, splicing and post-transcriptional modifications are done, but from the nucleus to the cytoplasm, it is not getting transported. Unless it goes to the ribosome in the cytoplasm or on the ER, it will not get translated. So that way also you can stop. At the level of translation, it has migrated from the nucleus to the cytoplasm, gone to the ribosome also, but over there translation is delayed, it is halted. That also can happen. At various levels in eukaryotes, you can see how the gene expression is regulated. Gene expression regulation in eukaryotes is a little complicated, but in prokaryotes is easier to understand. For that, we have an example of lac operon. Lac here is not money, lakhs of rupees. Lac here is short for lactose. Lactose is a disaccharide, a type of carbohydrate that you find in cells. You find it in milk. So basically, this lac operon was studied in E. coli bacteria. I'll tell you what exactly this is. This was elucidated by two people, Jacob and Monod. What is an operon here? I'm telling you this is about prokaryotes. So obviously the structural gene is going to be polycistronic. And this polycistronic gene, this structural gene that is coding for more than one polypeptide, if this is controlled by a common promoter, it is called an operon. Not just the promoter, along with the promoter, there will be certain regulatory sequences as well. If a polycistronic structural gene, which is responsible for producing more than one polypeptide, has a common promoter and regulatory sequences, we call it an operon. So in lac operon, we are looking at the structural genes that will synthesize enzymes required for the metabolism of lactose. For lactose to be broken down and used up, whatever genes are required, they are controlled by a common promoter and regulatory sequences. That is why it's called lac operon. So here's what the lac operon looks like. You have your promoter here. Okay. You have structural genes. There are three. It is polycistronic, responsible for the production of three enzymes: lac Z, lac Y, lac A. These structural genes will produce the enzymes beta-galactosidase, permease and transacetylase. I told you this is about metabolism of lactose. They should produce these enzymes only when the medium in which bacteria is growing has lactose. These enzymes are required for the breakdown of lactose. If there is no lactose in the medium, why should these enzymes be produced? Their job is to break down lactose. If lactose itself is not there, what is the point of the production of these enzymes? In such cases, these genes have to be switched off. They have to be turned off. Alright. Now, you know how most organisms prefer glucose to be the substrate for energy requirement. In cases when around E. coli bacteria or in the medium in which E. coli bacterium does not have glucose and it has lactose, only in such cases this enzyme has to be produced. If the medium has both glucose and lactose, even then you will not need the enzyme because glucose is preferred. In a case where you don't have glucose but you have lactose, these enzymes will be required. In all other cases, it has to be switched off. Alright. Now, over here, in the first case, lactose is absent. Lactose over here is the inducer. Okay. Lactose is absent. Before the promoter of these structural genes, you have another gene called the repressor gene represented by lowercase I. This repressor gene will have its own promoter. The repressor gene will produce the repressor mRNA. This repressor mRNA will produce the repressor protein. Okay. Now, in between the promoter and the structural gene of lactoperon, you have a regulatory sequence that is called the operator. This operator itself will act as the switch. This repressor has the ability to go and bind to the operator. Whenever there is no lactose, the repressor that is always going to be synthesized constitutively, this repressor will go and bind with the operator. Once it is bound to the operator, remember for transcription what do we need? We need RNA polymerase. RNA polymerase along with sigma factor will recognize the promoter, go on and form the RNA, and at the end it will get terminated. If the operator is bound to the repressor, even though RNA polymerase comes and binds to the promoter here, it will not be able to transcribe the structural gene. If transcription itself is not taking place, then translation will not happen, enzymes will not be produced. So in the absence of lactose, this repressor will turn off the operon and make sure that these enzymes are not going to be produced. What would happen if lactose is there? Lactose will bind to the repressor if lactose is there. The binding of lactose to the repressor will inactivate the repressor in such a way that it will no longer be able to bind to the promoter. Just when these enzymes are required, when lactose is there, lactose itself will inactivate the repressor. Now that there is nothing blocking the operator, the RNA polymerase will bind to the promoter here, transcribe the mRNA, polycistronic mRNA. From that we will get the three enzymes: beta-galactosidase, permease and transacetylase. Beta-galactosidase is the enzyme that will break down lactose into glucose and galactose. Permease is the enzyme that will allow the membrane of the bacterial cell, it will make it more permeable to lactose, so that lactose from the surrounding medium can enter into the bacterial cell. Transacetylase is an enzyme that is required to transfer acetyl group between two molecules. All of these are required for the metabolism of lactose in E. coli. So these should be produced only when there is lactose, otherwise it's a waste. So whenever there is lactose, that itself will inactivate the repressor and ensure that these are produced. Because lactose is inducing the production of these enzymes, we call lactose an inducer in this case. Okay, so this is how one of the ways in which gene expression can be regulated. Alright, I hope this is clear. Now let's learn about Human Genome Project. Human Genome Project was a mega project that was launched in the 1990s, I think. Yeah, 1990. And this project took 13 years to get completed. It was coordinated by many institutions like the US Department of Energy, National Institute of Health Sciences, and health partners include Wellcome Trust, Japan, France, Germany, China. Many countries were involved in this project. What basically was the aim of this project is to identify the sequence of the entire human genome. One organism, humans, one cells, one set of genetic information, just half, that is one set, one genome. For it to be sequenced entirely, it took 13 long years. Okay. So the goals over here was to identify approximately 20,000 to 25,000 genes that they estimated to be present in humans. All of the 3 billion chemical base pairs of DNA. What exactly is the sequence of base pairs? Whatever information you get, that has to be stored in databases. If you have to analyze the information that you have, you should have certain tools to analyze that. So the idea was to improve these tools that are available for data analysis. If possible, related technologies had to be transferred to other industries, and if at all any ethical, legal or social issues arise, then to address those issues were the goals of HGP. So how exactly did they accomplish this? There are two methods. One is express sequence tags and the other one is sequence annotation. In express sequence tags, you know how in humans, which are eukaryotes, there are only certain regions which will code for proteins, majority will not. So they only focused on sequencing the regions or the segments of DNA which had information. So that is why it's called express sequence tags. Excuse me. And the other one is called sequence annotation. Over here, they took a blind approach of sequencing the entire genome and later identifying regions which had information and annotating them. Okay. So what they did was, in order to sequence it, they took one cell. From one cell, they took one set of chromosomes, just the genome, and then they sequenced it by making use of computer programs. For sequence annotation, they introduced these fragments of DNA. They cut DNA into fragments and introduced them into hosts for cloning and making more copies. The hosts that were most commonly used are bacterial artificial chromosomes and yeast artificial chromosomes. More about what vectors are, how exactly they function, you learn in the chapter biotechnology. Okay, so at the end of 13 years, we were able to figure out the sequence of genome from one cell of one human. So what did we get to know about the human genome? Human genome contains 3,164.7 million base pairs. An average gene consists about 3,000 bases. The largest known human gene is the dystrophin gene that contains 2.4 million base pairs. The total number of genes is estimated at 30,000. Earlier, people thought 80 to 140,000 genes are present. About 99.9% of nucleotide bases are exactly the same in all people. The variation between two people is in less than 1%. Of all the genes we've known, more than 50% of the genes, discovered genes, we don't yet know their functions. And of all of these base pairs, less than 2% codes for proteins. In the other regions, you do not have any information. These are the things that we got to know from the Human Genome Project. So yeah, these are some things that you have to remember. Also, in the human genome, we have repetitive sequences, a lot of repetitive sequences, which means the same nucleotides are repeated multiple times. It could be a single nucleotide, it could be 4 nucleotides, it could be 6 nucleotides. The same sequence is repeated multiple times. That's called repetitive sequences. Sometimes they are repeated 100 to 1000 times. They do not have any functions in coding. They do not code for any amino acids or proteins, but they do shed light on the structure of chromosomes and the dynamics and evolution. The highest number of genes are found in chromosome 1. It has 2,968 genes. The least number of genes are found in chromosome Y in males. There are just 231 genes. There are 1.4 million locations in the human genome where there is difference only in a single base. They are called SNPs. Single nucleotide polymorphisms, they are pronounced as SNPs. They are found in 1.4 million locations. So that's about the salient features of the Human Genome Project. Now we have arrived at the last concept of this chapter, which is DNA fingerprinting. I'm sure you've heard of DNA fingerprinting when you watch any crime series. In forensics, they use DNA fingerprinting to test for paternity. They use DNA fingerprinting. So how exactly is this done? This DNA fingerprint has nothing to do with the fingerprint over here. Just like we say our fingerprint is unique, our DNA is also unique. Unless two individuals are identical twins, you will not find same DNA in two people. That's what makes it unique, okay? So what basically is done during DNA fingerprinting is you isolate the DNA. If it's a crime scene, you isolate it from any living cell. It could be blood, it could be hair, it could be semen, it could be anything. So you isolate DNA, you take this DNA and digest it. Digest it means make use of enzymes to cut the DNA into fragments, because DNA is a very long, what do we say, molecule, right? So you cut it. In order to cut it, you make use of enzymes called restriction endonucleases. Don't worry much about it now. In detail about restriction endonucleases, you learn in biotechnology chapter. So these enzymes are used to break the long molecule of DNA into shorter fragments. Later, based on the density or length of the fragment, they are separated. Okay, so they are separated by a process called gel electrophoresis. Now, how does it differ between two individuals? Like we already said, 99.9% of sequences are similar in most individuals, right? There is variation, little variation. And there is a lot of repetitive sequences that we find in our human genome. So what happens here is, because of mutations over the course of time, different people would have had slight modifications in the genome. That is what is the basis for DNA replication. The polymorphism or the differences that exist between populations, individuals belonging to the same population, is what acts like the principle behind DNA replication. Like I already mentioned, we have many repetitive sequences. They are called, they are classified into different types: microsatellites, mini satellites, and all of that. There is a type of mini satellite called VNTR. Okay, so VNTR stands for variable number of tandem repeats. For example, let's say CAT. These three nucleotides are repeated multiple times at one point of my DNA. The same CAT in my DNA, it's repeated 30 times, in your DNA, it's repeated 40 times, in your friend's DNA, it's repeated 400 times. So the letters are the same, the word is the same, but the number of times it's repeated is variable. Because of that, when I cut my DNA, I'll get a short fragment. When I cut the DNA of a person that has it repeated 40 times, it'll be slightly longer. When I cut the DNA of a person that has it repeated 400 times, it'll be much longer. So we are using the same enzymes to cut the DNA of two individuals. In me, it will be short. In the person that has it repeated 400 times, it will be longer. So after that, when you try to separate it based on the size, the kind of bands that I get will be very different from the kind of bands that another person gets. Two people can have the same number of repeats for one word. Okay, so in me, cat could be repeated 30 times, and another person also it could be 30 times. But there is another such tandem repeat that can be variable. We have many such. I'm just giving you one example. So CAT can be the same, but there could be TAC repeated 82 times in me, and TAC could be repeated 30 times in another person. So there are many such tandem repeats that we see in DNA that varies from one individual to another. When you cut the DNA into fragments, so you're adding restriction endonucleases into my DNA sample and restriction endonucleases to the DNA sample of another person. So when you add that, you're going to get fragments of different lengths. Later in the next step, gel electrophoresis is performed. Exactly how it's done, you learn in biotechnology. It will separate the fragments of DNA based on their length. Because mine is shorter, it will get separated sooner. Because another person's is longer, it might get separated at another point. Okay, so the banding pattern that we are going to get will not be the same. After gel electrophoresis is performed, the DNA would have formed some banding pattern. That DNA has to be transferred to a nylon membrane or a nitrocellulose paper, and that is done by a process called southern blotting. After that, the double-stranded DNA is made to form single-stranded DNA. Since I am the one who cut the DNA, I know it is cat that I was looking for. So complementary to cat, we have GTA. I will synthesize probes, radioactive probes of GTA, and allow that to bind with this. In whichever organism, in whichever sample CAT is there it will go and bind. Okay, so remember this is radioactive. So the single-stranded DNA is hybridized using radioactive VNTR probes. Later, if you keep this against an X-ray film, you will get banding pattern on the X-ray film as well. That is called the autoradiograph or the autoradiogram. And from that you will get your DNA fingerprint basically. Okay. So here's how it looks like. You extract it, you cut it using restriction enzymes, you get fragments, you separate that using gel electrophoresis, you will get bands like this over here. You transfer this onto a membrane, it could be a nitrocellulose paper or nylon membrane. The DNA fragments here, because of capillarity, will move, will get adhered onto this paper. You make it single-stranded. You incubate that with radio-labeled VNTR probes. If at all that sequence is present, it will go bind to it. After you keep it against an X-ray film, you will get your autoradiogram. This is your DNA fingerprint. So if in a crime scene you've got some sample, like a blood sample or a hair sample, and you have two suspects, you can collect DNA from them. You can do the DNA, you can collect the DNA from your hair follicle. You compare it. If the DNA fingerprint from the hair matches one of your suspects, then you know who was at the crime scene. If it is for paternity testing, exactly half, 50% of the bands are going to match. So that way you can tell who is the parent, who is the father of the child. Okay, so that is DNA fingerprinting for you. So with this, we have come to the end of today's lecture. It was lengthy, I understand, but I hope every concept that we've discussed today is crystal clear for you all. With this, I will sign off from today's session. Until I see you next, take care of yourselves, stay happy and keep smiling.